Thank you so much for the kind introduction and for the invitation. I'm very much looking forward to talk a little bit about very early child development today, and especially the role that social rhythms and biobehavioral synchrony play in early human development. And yeah, just I prepared a presentation with a little bit of theoretical background and some studies, but please do let me know if there are any things that are unclear, or if any questions in between, that's fine. And also, if I'm talking too long, just yeah, give me a little hint and I will try to speed up or wrap it up. Okay, so just up front, if you're interested in learning a little bit more about our research, here are our social media channels and also homepage. And yeah, let's dive in. So the first thing I always like to point out is that once you start looking for rhythms in the environment and in the body, actually, you will cannot stop noticing that rhythms really abound. And especially if you look at how caregivers interact with their pre-verbal children, with their babies very early on, you will recognize that these interactions are inherently very rhythmic. So there's a specific rhythmicity in infant-directed speech that I'm going to talk about. I'm going to talk about proto-conversations, so even before being able to speak, the conversations that caregivers have with their babies. There's nursery rhymes, of course, lots of musical interactions. There's also tactile rhythm in affectionate touch. And then infants, of course, they experience being carried around, being rocked. And also there's lots of interactive games that also have a certain rhythmicity. And once you kind of scale up a little bit, also there's regularities in the daily routines that infants experience in their very early environments and early lives. And yeah, once you start noticing all of these rhythms, you might start thinking about why are they there? That's probably not completely coincidental. I think we've made this argument before that these rhythms are really useful for babies because babies are very helpless when they're born, but these rhythms make the world more predictable. Infant brains are very sensitive. They pick up on regularities very well. So these rhythms are really helpful in making the world more predictable and also in achieving caregiver and infant interpersonal synchrony. And I'm going to talk about both a little bit. What do I mean with predictable world for infants? We can actually show that infants very, very early on form expectations also about their social interactions. And a very striking example for that, you might have come across that maybe if you've studied psychology, for instance. A very striking example is the still face experiment. So we don't have the sound now, so I'm just going to walk you through it a little bit. I think the visuals will suffice to kind of carry the message. So what you see here is an example of the still face experiment. This is a mother interacting, talking with her child. You see there's lots of back and forth, lots of positive affect in the interaction. The baby points somewhere, the mom follows. So this is a baby that's almost a year old. Here we have an example of a little game where both end up laughing. And now the mom is instructed to assume a still face. So what you're going to see now is the mom doing as she was instructed by the experimenter, just not doing anything anymore, just looking at the baby. And you will see that the baby notices this quite quickly. First, tries to kind of re-engage the mom in the interaction by pointing here, look there, that kind of worked before, but trying to grasp her, really trying to make her respond. And yeah, the mom doing as you see, it's kind of getting hard for her as well. And now you see that the baby is starting to stress out. Yeah, so this is really something unexpected for the baby. This is really starting to be stressful. Now I'm kind of glad we don't have the sound because it's also not nice to kind of listen to. The baby is really visibly and audibly getting more distressed by this interaction. these experiments don't last very long, so I'm just going to show you now kind of the resolution. So the mom is back, and now very quickly the baby is also back and very happy again. And this is called the reunion phase, where we usually see a repair of this broken down interaction. But what that shows us, so this baby was already a year old, but we can see these kind of responses to the still phase already by 6 weeks, 8 weeks of age. So very early on, infants get very distressed when they don't get a response from their caregiver. But we don't see it at birth, yeah? So this is really something that's learned quite quickly, where infants seem to realize that through their own communicative actions, they can elicit responses, and they rely on that. Yeah, they start to rely on that. So the kind of reunion phase and also the normal play phase at the beginning were very nice examples also for kind of the interactive communicative flow that infants are used to experience with their caregivers. And what has been shown in seminal research by Ruth Feldman and her lab is that these rhythmic interactions induce behavioral and physiological synchrony between caregiver and infant. So we saw some examples of that also in the video, when both were sharing smiles, sharing laughter, positive affect. And what she showed in her research with her group is that this behavioral synchrony is also linked to physiological synchrony. These are closely related. For instance, she showed that by measuring heart rate coupling, yeah, so the heart rate of the infant and of the mom. She also showed that in endocrine fit. So this is an example of oxytocin release correlation between mother and child. And she showed that these levels of synchrony are actually also related to the parent-infant bond. So they relate to later attachment security, for instance. And it has been argued quite convincingly, I think, that this is really critical, especially for young infants that rely on co-regulation. So they are not yet able to stay in homeostasis. So in all the physical needs need to be met by the caregiver, but also their emotional needs. Stress regulation is something that infants are very bad at the beginning. They really need a sensitive caregiver to kind of pick up on their stress response and then kind of get it down again. And this might even be quite important for infants' formations of model of the self and of the other. Now, what has been researched much less, so I call it underexplored questions, is whether these communicative rhythms and synchrony, whether this also plays a role for social learning, for communication, more like the cognitive aspects of development, and whether there is a role also for neurosynchrony, so synchrony of brain rhythms. Now, why could that be something interesting to study? I'm guessing that a lot of the audience members are kind of already familiar with the concept of neurosynchrony or hyperscanning, but I'm just going to start from a very basic level, thinking about this. And the first time I started thinking about this or hearing about this was in research on speech tracking. Yeah, so entrainment of neural oscillations to speech. This is where it comes from. And the basic premise is quite simple. It is admittedly also a simplified kind of illustration of what's going on there. You see brain oscillations as kind of fluctuations in the level of excitability of neuronal populations. Yeah, so we know that brain rhythms aren't steady; they oscillate, and this also relates to processing efficiency. So the very same physical stimulus can occur at a phase in the neural oscillation when processing efficiency is very low, and we might not even notice it, or it might occur at a time when processing efficiency is high, and we can perceive it very well. Now, if we take this kind of basic idea and apply to rhythmic stimulus, we can imagine a situation where this rhythmic, this regular stimulus always occurs at a time when processing efficiency is low, or it can be optimally aligned to the brain rhythm. Yeah, and this is something that we see again first to my knowledge, has been shown, at least in ecological context, with speech. Speech is, of course, not a perfectly periodic signal, but it's sometimes called quasi-rhythmic. Yeah, it entails rhythms. And it has been shown that the brain rhythms basically track the speech rhythms. And if we take this logic kind of one step further to two interacting partners, and both basically align to each other's communicative rhythms, align their own speech, perhaps, and their brain rhythms to each other, this is how we can, in principle, achieve something like neural synchrony. Yeah? So basically the alignment of brain rhythms from both partners. And I already mentioned that infant brains pick up on regularities and pick up on rhythms very well. They're very sensitive. This also applies to adult brains. Actually, our brain activity entrains to sensory rhythms. And we argued in this paper in Scan some years ago that actually all kinds of rhythms are represented by our brain activity. Yeah, striatocortical loops represent any kind of repetitive interval sequence quite automatically. Yeah, in this illustration, you see this kind of notes. So would be an example for music, for instance. Yeah, but actually, when we look at the research, when we look at the studies, not all rhythms are treated equally, and this is where socio-emotional processes come into play. Supported by posterior superior temporal cortex, because these endow sequences with value, either motivating, synchronizing to these rhythms or not. So music, for instance, is much more effective in training our brain rhythms and also our motor rhythms, our whole bodies, than just, periodic tones that are boring and not so interesting. So attention also plays a role here. And we showed in this review paper that there's preferential entrainment, especially to rhythmic stimuli that are multimodal. Yeah, so if we have both the speech, for instance, and the mouth movements, and usually actually gestures are also in sync with that, that helps. Positive, yeah, if it's something that we like. Natural and complex, which I found initially surprising. I thought, oh, well, the more periodic and simple, the better. We should be able to entrain, but no, actually music, it's much better than just the tones. So we argue that communicative rhythms are actually ideal stimuli to induce mutual entrainment in an interaction and result in neurosynchronization. And this neurosynchrony, as argued before, shown before in speech tracking research, can then promote communication and interpersonal coordination. Because it also facilitates mutual prediction. It actually takes off some of the cognitive load. So to just kind of illustrate this, again, we can think of different pathways basically to synchronizing two brains or even more brains. One is within the diet, basically, through communicative rhythms that are directly exchanged between the two persons, a speaker producing a speech rhythm, the listener picking up these rhythms in their own brain activity, and this can, in a back and forth, result in synchrony. Other stimuli that are rhythmic in communication are biological motion, affectionate touch, laughter, something that we're currently studying. All of these communicative signals are rhythmic in nature. But then, of course, there can also be external rhythms jointly attending to something in the environment, which might also help us synchronize our brain activities. And here a classic example is music, as I mentioned before. But we can also think about joint visual attention, even shared narrative processing. I don't have so much time to go into that, but this could basically entail a triadic situation where two people engage together with something in the environment which helps them synchronize. And very often in real life, I think these are not mutually exclusive pathways to synchrony, but they actually they actually occur at the same time. Okay, so in order to study that in very early human development, we kind of have to adapt our paradigms and our research methods. It's not easy, I can tell you. Usually what you want to do, especially when studying infants, is have them sit maybe in front of a screen and entertain them, but have them move as little as possible because they tend to produce lots of artifacts, for instance, in the EEG. So what we've been trying to do is kind of take infant neuroscience a little bit in a more interactive direction. First, starting to measure infants' brain activities while they are engaged with an adult interactive partner in a real-life interaction, and then in more recent studies also to measure brain activities from the adult and the infant at the same time. So what we're trying to do here is capturing the dynamics of multimodal social interactions, because this is how infants in their real lives, how they actually learn and how they experience the world, not in front of a TV, at least not most of the time. So, and this brings me to the first set of studies I want to present to you. And I'm staying with the topic of tracking of speech and also of singing in naturalistic interactions. So the first modality I'm going to focus on is the auditory modality. So the speaker-listener coupling that I alluded to, that's been well researched in adult research. Is it something that we can see in infants? And this study has been done at the Max Planck Institute in Leipzig and was led by Katharina Mell. And she was interested in whether the infant brain would actually track speech and whether it would be able to, or would preferentially track infant-directed speech. Now, we know that infant-directed speech is something special. It's sometimes called motherese, and you probably intuitively know what it is. It's kind of high-pitched, more variable, more affectionate speech that adults, caregivers, but also other adults intuitively use when speaking to infants. Now, I'm often asked whether actually it makes sense to talk to infants that way. Wouldn't it be better to talk with them like adults because they should learn to speak properly? And I always say that, well, the research is very clear on that. Infants prefer infant-directed speech over adult-directed speech. They pay much more attention to it. They listen to it much longer. And it actually helps them in their language acquisition. So it helps them in word segmentation, kind of identifying words in the speech stream, word recognition, learning of new word-object association. So there's lots of benefits of talking to infants in this kind of exaggerated manner, in this affectionate manner. So it actually makes sense that usually adults do that intuitively. And we also knew from previous research that infants' brains track speech. They synchronize activity with amplitude modulations corresponding to the prosodic stress and also syllables and nursery rhymes. But this previous research was done with pre-recorded audio. So we were interested in real-life interaction, basically. So we looked at whether infants' brains would track speech in a naturalistic interaction with their own mother, and whether neural tracking would be facilitated specifically by prosodic stress or syllable information in infant-directed speech. So what we had moms do, so infants here were 9 months of age. We invited them to the lab, as you see here on the picture. And then we just asked moms to show their babies these novel, unfamiliar objects and talk about the objects as they would usually do, when they're showing something to their infant. Or we asked them to talk about the objects in a kind of, in a manner like they were talking to an adult. So we did not instruct them to talk in infant-directed speech, just said, either talk to your baby like you would usually do, or as if you are talking to an adult. It was a within-subject design. And now, I would, now the audio would be nice, but I guess you get the idea.
Was ist denn das? Huh? Ein blaues Ding, eine Krake? Ist das vielleicht eine Krake?
So the mom is basically talking about this kind of object, what could it be? And as I said, intuitively, kind of spontaneously actually engaging in infant-directed speech. And then in the other condition, basically just talking about the object like she would talk with an adult. So what we found here in first of all, analyzing the acoustic properties of the speech is that indeed, even without explicit instruction, the moms did engage in infant-directed speech, like prototypical infant-directed speech, in the kind of normal talk to your infant condition, which means it had higher pitch range, and especially the amplitude modulations in the stress rate were increased for infant-directed speech compared to adult-directed speech. The syllables were kind of not different, not significantly different, the modulations in the speech amplitude for the syllables. And what we did next is then look at the speech-brain coherence, specifically in the prosodic stress rate. So these would be the kind of slow changes that go along with the speech prosody, the melody of the speech, and then the faster syllable rate, which is usually 4 to 6 hertz. Yeah, so 4 to 6 syllables per second is the normal speech range, and that was not different here for the infant-directed speech. Okay, so what we do is we take the EEG from the infant and the speech from the mom. We especially extract the envelope from the speech, and then we compute the coherence between the EEG signal and the speech envelope in these two frequency ranges. And what we found was that indeed the infant brain tracks speech, both infant-directed speech and adult-directed speech, but it's better at tracking infant-directed speech, especially in the prosodic stress rate. Yeah. So for the syllable rate, there was significant coherence compared to chance level, basically, or control condition. But in the stress rate, we see a clear advantage of the infant-directed speech. So we can conclude from this study that the infant brain does track natural speech that's kind of spontaneously occurring in an interaction. So yeah, my guess is this is also something that happens in their everyday life. And we can say that the benefits of infant-directed speech stem especially from the enhanced prosodic stress. So it seems like mothers actually modulate their speech in a way that it is actually better trackable by the infant brain. Yeah, so the infant brain has an easier job tracking the speech rhythm. Yeah, but what caregivers also often do is not just talk to their infants, but also sing to their infants. So this is something that we looked at in another study. We looked at infant-directed singing, also again in a naturalistic interaction. Based on research showing that caregivers sing spontaneously to their infants, and they seem to use kind of two different types of song, play songs and lullabies that differ in their acoustic qualities again. Yeah, play songs are kind of more engaging, more variable. Lullabies are kind of lower in pitch, slower, and also less variable. So what we did here is, again, we measured the infant's EEG. Those were 7-month-olds, and we had their mothers sing different songs. And this time we used a little different analysis approach. We used temporal response function. So what we do is basically we train a model based on 80% of the data, to kind of link the stimulus, in this case, the maternal singing, and the EEG response of the infant. And then we test whether that model actually can predict the infant EEG based on the stimulus, so based on 20% of the singing. And then we can basically correlate the predicted EEG response and the real EEG response that we collected and see how accurately the model can predict the infant EEG response. And as in all of the studies I'm going to present, I'm not always going to say exactly what we did, but we always ran control analysis. In this case, we used randomly shifted EEG data and looked at the correlation with the predicted response from the individual model. And this is what we did, what we found. So again, we do see a significant basically tracking of the singing in this case, significantly higher prediction accuracy for the true compared to the shifted EEG data, and actually higher accuracy for the lullabies than the play songs, especially for the songs that were sung more slowly. So again, we didn't instruct the moms a lot. So we basically asked them to sing a kind of prototypical German play song or a prototypical German lullaby. And so again, we had lots of variability actually in the production, in the performance. But we saw that those moms that sang the lullabies, especially slow, here the baby's brains tracked these songs very well. But at the same time, we saw that play songs elicited more rhythmic movements, not very surprising. But only infants' neural and behavioral coordination with the more variable play songs, which were harder to track overall, predicted their vocabulary size at 20 months. So this is basically when we, a year or so after the testing in the lab, we asked the moms again to judge the vocabulary size of the infants, and infants' tracking of the play songs actually predicted this. So the infant brain tracks natural infant-directed singing, not only speech, but also singing, and the slower tempo of lullabies promotes neural tracking. But the tracking of the play songs, which is harder generally, might be more predictive of infant language development. Okay, so those were two examples of communicative cues in the auditory domain, but we also ran some studies focusing more on visual communicative signals, and especially here I brought you one study on joint attention that's I think quite interesting. And what we looked at here were the social origins of cultural differences in visual attention. What do I mean? This is a study that was run by Anna Banki, a PhD student in my lab, and she was interested in cultural differences in context sensitivity. This is also something that you might have heard about before, especially if you have background in psychology. It's a well-replicated, well-shown effect that in Western cultures, attention is focused mostly on central objects or images or scenes. Yeah, so basically, when you show someone from a Western European or Northern American culture a picture, what they will pay attention to, what they will point out, what they will remember later on, is usually what's at the center of the screen. And in Eastern cultures, Asian cultures, there's much more description and memorizing also of details that might be in the background of the scene. Again, quite well replicated findings, and we don't know actually much about their ontogenetic origins. So we know that this is something that we see quite robustly in adults. So in this study in parents, for instance, we already see it in children, 7 to 8 years of age. Yeah, so those were Canadian children and Japanese children, and they showed this effect quite nicely. So here what you see is accounts of the reference to the background, which is one of these measures where we see this difference in context sensitivity. But if we see it in children, where does it come from? So what we reasoned is that it's, well, at least one kind of explanation or mechanism how that might emerge might be social interaction, and basically the socialization within caregiver-child interactions in very early development. So what we did here is we worked with 12-month-olds and their moms. We had one sample that we tested in Austria, in Vienna, and one sample that we tested in Japan, in Kyoto. And what we did is we showed infants these images that had a clear kind of object in the center and a clear background. And we presented those objects flickering at different frequencies. It's often called frequency tagging, this general approach, where basically you flicker on off images on the screen. yeah, at a particular frequency. So for instance, here we might have flickered the clownfish at 8.5 hertz, but the background, the corals, at 5.66 hertz. Yeah, we counterbalanced what frequency was used for the object or the background. And what we see then is, especially in visual regions, so under occipital electrodes, what's called a steady state visual evoked potential. So we see basically activity increase at exactly the presentation frequency, which is very useful, especially if you want to look at kind of, what infants who cannot yet speak, what they pay attention to on a screen, for instance. Yeah. So we flickered these objects and backgrounds at different frequencies, and then we can really quantify nicely what the participants pay attention to. So we did that in a pre-phase. Then we had an interaction phase where we just asked moms to point out something on the screen to their infant. That was it, basically, yeah, as they would usually do again. So quite naturalistic. And then we had another phase where we just showed the images flickering. So the first thing we looked at was what the moms actually chose to point out for their infants. And that's the first really interesting result in this study. Moms from Vienna pointed to, a lot of moms from Vienna pointed to the object 100% of the time. So we didn't tell them to point to the object at the center, they just did, like a lot of them all of the time. And this was not the case to that extent in Kyoto. In Kyoto, we get much more variability. So a lot of the moms actually pointed to the background 50% of the time or more. So indeed, there seems to be something to this idea that this is something that's transmitted socially in very early social interactions. Yeah, so some of the babies in Vienna, they are always pointed, their attention is always pointed to central focal objects. Now, what did the infant EEG tell us? Spontaneous maternal guidance of infant visual attention is culture specific. What did the EEG tell us? So what we see here is now what we call the object score. So in terms of SNR, it's also a nice thing about these flickering paradigms that you can actually nicely compute SNR because you know exactly what the signal of interest is because it's the stimulation frequency. Yeah, so a lot of the times we have difficulties with EEG measures in infants, ERPs, and all of these because there's lots of noise, signal to noise ratio isn't so good, but with these flickering paradigms, that works very, very well. And what we found here is that infants in Vienna, even at the pre-phase, even before the interaction, paid much more attention to the focal objects. Yeah, so their SNRs, their brain activities at the frequency of the object in the center was much higher than for the background. And it's almost the opposite for infants in Kyoto. Yeah, so they pay much more attention also to the background. So we do see cultural differences in visual attention already by 1 year of age, already in the pre-phase of the experiment before the interaction. So this is something that they've learned before, I would guess. Yeah, they've already had a couple of months of experience in joint attention, in having their attention pointed to different things. And I think what we see in the study in the interaction phase is an example, a snapshot basically of their daily experiences, where their attention is socially guided in different ways in Japan compared to Austria. Okay, so yeah, that was auditory visual. Let's move to another communication channel that's really, really important for infants, and it's touch. So, and now we're actually making a switch to this kind of third third kind of paradigm where we're measuring brain activities not only from infants, but also from caregivers. And now we're also kind of necessarily switching to FNIRS because it's less sensitive to motion artifacts than EEG that we used in the study so far. Okay, this is a study that was led by Jin Nguyen. who used to be a doctoral student in my lab, is now a postdoc in Rome. And what she did is she was interested in neurosynchrony in mother-infant diets. So she measured their brain activity with FNIRS. And she also at the same time measured ECG. And she performed video recordings of the diets in different situations. In those different situations, the babies were very young, 4 to 6 months old. And the different situations that we created were these here. We had a proximal watching condition where the baby was held by the mom and they were both looking at something on a tablet, aquarium video with fish. We had a distal watching condition where both were also watching something on a tablet, but no touch this time, no bodily contact. And then we had a free play interaction where basically touch was allowed. Yeah, so here we had varying levels of spontaneous touch between the mother and the infant. We just asked them not to sing to kind of control for this level of rhythmicity. And we computed neurosynchrony between the brain activities of the babies and the moms by way of wavelet transform coherence, which is the most commonly used measure of neurosynchrony in FNU studies. So we used that as well. And the first thing we saw, so of course we lost some data, so now we're looking at data from 69 diets, was that the coherence between infants and mothers in neurosynchrony was highest in the free play condition compared to the proximal watching condition. And it was higher in the free play and proximal watching condition compared to the distal watching condition. So the conditions, the two conditions where we allow touch at the higher levels of synchrony, basically. And bodily contact had an immediate effect on neurosynchrony. Yeah, so higher neurosynchrony was found during the proximal watching condition where the mom held the baby compared to the distal passive watching condition. Now we kind of zoomed in the free play interaction and looked at what's happening there. Again, this is a nice thing about these paradigms. There's lots of natural variation in what the moms did and what the infants did. And the one thing we noticed is that higher or longer durations of affectionate touch were related to increased neurosynchrony, but longer durations of stimulating touch, so tickling, poking, these kinds of things, were related to decreased neurosynchrony. So not all sorts of touch basically had the same effect. And we think that, well, high levels of affectionate touch, like soft stroking, this might be something that's really calming for this might be have some co-regulatory function. Whereas stimulating touch might be something that moms actually try to re-engage their infants in the interaction. And it might be kind of perceived as even intrusive and might then not be indicative of a very kind of aligned synchronous interaction. I briefly mentioned we also looked at ECG in this study. So we also measured respiratory sinus arrhythmia, and here we used cross-recurrence quantification analysis to look at the recurrence rates, and we found that, and surprisingly for me, we did not find a difference in physiological synchrony between the distal watching and the proximal watching, but we found the highest level of physiological synchrony in the face-to-face free play interaction. And we actually saw that physiological synchrony was related to infant affect. So the happier the baby, the less synchrony we found. Once the babies were stressed, so when they showed negative affect, this is when we found higher levels of physiological synchrony. And if you think about that, that makes a lot of sense because, well, we had this lab environment, the baby was getting unhappy, the baby was getting stressed. So this would stress the mom as well, right? And actually it aligns nicely with studies using day-long recordings of mothers and infants, showing that physiological synchrony occurs especially at times of peak stress for the babies. So Sam was ran some studies on that and calling it the there when you need me. Moms do not over the whole day all the time kind of synchronize with their babies. It's probably also that wouldn't be functional because babies, as I said, aren't so regulated. Yeah, so there's lots of variation, and moms don't usually, if it's not needed, don't synchronize their own physiology to their baby. But when the baby is stressed, this is when they also get basically alarmed and also we see this synchronous physiological response. We found no correlations with touch and no correlations with neurosynchrony, which I think is very interesting because sometimes synchrony in different domains and different modalities like neural, behavioral, physiological is all kind of mentioned in the same way or perhaps interpreted in the same way. But here I think we see some distinction. Yeah. So when we look at physiological and neurosynchrony, they are both enhanced in the free play interaction, in the face-to-face interaction, but we see different behavioral correlates, and this might reflect the fact that we might have different functions for these different levels of synchronization between caregivers and infants. Okay, so let's turn to it's not really a modality, but the last kind of piece of the puzzle I'm going to present today, looking at temporal aspects of the interaction. So what I became very interested in is turn-taking in conversations, in verbal conversations. I think it's fascinating because it's so quick. Yeah. So when we have a normal conversation with somebody, and it's also across different cultures, on average, it takes us 200 milliseconds to respond to each other. Compared to when you're just, you are asked to produce a label for whatever tree or dog or whatever, that takes you a second almost, yeah, to retrieve the label. So what I think that shows is that during a verbal conversation, we're constantly predicting each other and constantly kind of predicting when it's our turn, what's going to be kind of the final words of the statement of the other person, and kind of preparing our response already while the other person is speaking. So this requires a high level of mutual engagement and mutual prediction. And it has actually been argued that faster response times and more frequent turn-taking could be something like an honest signal of social connection, because it's impossible to fake. Yeah. So when you're talking with somebody and the conversation isn't flowing, it's kind of, there's interruptions and it's kind of awkward. You cannot fake it. Yeah? You cannot fake a social connection in that way. It's even perceivable to third-party kind of observers of the conversation. And now the fascinating thing is that the temporal structure in pre-verbal proto-conversation is very similar to later verbal conversations. So I have this cute video, won't play it now because we don't have the sound, but basically it's very cute. The baby is pointing to something and kind of babbling, and the dad is responding to the baby in a way like there was a real conversation. And then the baby turns, like it goes back and forth between the two as if it were a verbal conversation. It's very cute, and it's something that we see occur spontaneously between caregivers and their infants very early on. So at 3-4 months, these interactions are usually, yeah, they look like conversations. So, and this precise timing likely depends again on the synchronization of endogenous oscillators in both conversation partners. Yeah, it would be very hard to achieve this level of flow without kind of synchronization. So what we did is we tested again 4 to 6 months old babies and their moms in free play interactions, and we coded their vocal turn taking. And we assessed brain activities with FNIRS, hyposcanning. And the first thing we saw, as we had expected, exactly that more frequent turns were related to higher neurosynchrony, especially in the medial prefrontal cortex, and specifically, interestingly, in the early phases of the dyadic exchange. So indeed, there seems to be something to the idea that we need to have kind of our neural oscillators aligned to some degree to achieve this level of turn-taking, vocal turn-taking in this case. And again, we looked at infants' vocabulary size later on, some 20 months, 18 months later. And we saw that more frequent dyadic turns were related to, positively related to the size of infants, expressive vocabulary. So those infants that experienced this flow in the turn-taking in their proto-conversations also had bigger vocabularies later on. And we think what that shows is that turn-taking in mother-infant diets already relies on a high level of mutual intention and engagement, and that this could actually be very helpful in infants' language acquisition, which is in line with an interactionist view of early language acquisition that posits that it's not like the infant brain is not a sponge just passively receiving input, but it's actually the acquisition of language is actually highly dependent on the infant being active participant in this process and also experiencing responses to their own vocalizations, which we saw nicely in this study. Okay, so yeah, looking at the time, just to kind of summarize the studies that I've shown you. What we see across the board is that interpersonal neurosynchrony emerges spontaneously, and especially during active mutual engagement in parents and infants. So we don't see it as much when, for instance, mother and infant are just watching something. Yeah, they're just watching something on a tablet, a video. We don't see this neurosynchronization to the same extent compared to a real face-to-face mutual engagement. And this aligns with our work with preschoolers that we'd done previously, where we also found that mother-child neurosynchrony is related to behavioral reciprocity. So here we ran some studies, for instance, having preschoolers and their moms complete puzzles together, which were quite challenging. And we saw, again, a range of kind of different behaviors across the diets, because it's, again, a quite naturalistic task. And we showed that the more mother and child in this task actually respond to each other, the higher the level of neurosynchrony. And the more puzzles they were actually able to solve together. And we also showed in preschoolers and their mothers that there's a positive correlation between neurosynchrony and turn-taking. So now we're talking about real verbal turn-taking in real conversations, which we found to be related to increases in neural synchrony over time. Yeah? So the more turn-taking in the diet, the higher the level of neurosynchrony over time. Okay, but what can we basically take away from that? So we see that neurosynchrony especially is linked to interactional outcomes, positive outcomes in all of the studies that we conducted so far. And these are both kind of proximate outcomes like collaborative success in the puzzle game, but also some long-term outcomes like child language development, vocabulary size, and these things. We also looked at attachment quality and showed some links with that. So kind of really long-term effects of these early synchronized interactions. And some of these were actually not identifiable based on the behavioral coding, fine-grained behavioral coding that we did on the videos of these short in-lab interactions, at least. So what we could see here is that neurosynchrony might be a potentially useful biomarker. Potentially useful because we're not yet there at the point where we can say, oh, it's really something that we can use in diagnostics. This is not the case, but it seems to tell us something about the quality of the interaction, which is related to meaningful things. like language outcome or attachment. But in terms of kind of understanding what's going on in the interaction, we have a limitation. Because in all of our studies, what we're doing is basically we are looking at correlations between the neurosynchrony and what's going on the behavioral level or the physiological level. So we cannot really conclude whether neurosynchrony is like a mechanism that underlies social interactions or these positive aspects of social interactions, because we run into a sort of hen-egg problem, right? So is it the neurosynchrony that kind of allows us to have these nice fluid interactions, or is it because we have these nice interactions that we see a higher level of neurosynchronization? So we cannot solve this with this sort of research. But we can test some hypotheses, I think, that can inform a mechanistic understanding of social interactions. One thing that I showed you today is by really studying neural tracking of communicative rhythms, because I think this is really essential to understanding how neurosynchrony emerges in real-life interactions. Yeah, I showed you examples of speech, of music, of singing. Another project that we're running right now is looking at laughter, which is also a very positive, complex, natural rhythm that we produce in social interactions. And what we can also do is we can modulate neural oscillations, and we can modulate synchrony experimentally. For instance, through rhythmic stimulation. So I showed you an example of the flickering paradigm, which is a sort of rhythmic visual stimulation that can modulate neural oscillations. Or we can, modulate synchrony, for instance, through hyperscanning neurofeedback. This is something that we're starting right now. And we have a preprint of a kind of proof of concept paper where we ran some simulations with existing data, looking at kind of what level of data quality and what level of duration of recording do we need if we want to give a diet feedback on their neurosynchrony with fNews data that we collect during the interaction. So this is the paper by Kostwartz that's under revision. It's also out as a preprint. And this can help us understand better the functions or the benefits of synchrony. I'm a bit cautious with the word function. I don't know if this is what synchrony is for, so to say, but it could tell us something about the benefits of social synchrony and neurosynchrony in particular. So these are already some future directions. I think in the next steps, it would be really useful to understand better how different levels and modalities of interpersonal synchrony or alignment interact. So I talked a lot about brain synchrony today, but also talked about how this is related to synchrony on the behavioral level, things like joint action, joint attention. There's, again, lots of linguistic research on things like phonetic convergence or gesture coordination. I talked a little bit also about the physiological level, physiological co-regulation, heartbeat, breathing, endocrinology. And already in the first studies that we looked at where we used these multimodal recordings, we already see dissociations between these different levels of synchrony sometimes, at least between physiological co-regulation and brain synchrony. And then on a higher level, is something that I didn't talk about today. Conceptual alignment is, I think, something that's really, really interesting. Like, do we understand information in the same way? Do we interpret narratives in the same way or not? Which is also something that could be linked to brain synchrony. So this is something that I think we should understand much better, and also kind of combine different measures in the same studies to understand that better. I'm also interested in understanding how momentary interaction patterns relate to longer-term bonds. So the research that I showed you today, we always get these snapshots or these glimpses into how interactions unfold within a couple of minutes, 5-minute, 10-minute interactions. But I would be very interested to better understand how that relates to long-term bonds. Again, we're doing that a little bit by looking at attachment quality, but what I really think is needed here is longitudinal research, especially developmental longitudinal research. And this leads me to the third point: what is the role of intrapersonal and interpersonal coupling in child development? This we have barely started to tap into with our research, because again, we've only so far taken these snapshots but not looked at how synchrony across these different modalities evolves over early development. So just an example of a longitudinal study that I would like to run is looking at interpersonal neurosynchrony during joint book reading interactions between caregivers and children, and then linking interpersonal neurosynchrony during this exchange during the joint book reading to long-term shared representations, which in turn might predict how well caregiver and child synchronize in future interactions. So this would really require a longitudinal approach with different measurement time points across the first years. Okay, and then finally, if I have some more minutes, some future directions for the field in general. I think that we have the potential to gain really fresh insights for education, shifting towards teacher-learner dynamics as compared to focusing on the learner or the teacher or the environment. Yeah, really looking at again how teacher-learner dynamics unfold, both across kind of teaching interactions, but also across the long term. And it really kind of should push us to prioritize things like social well-being and sense of belonging during transitions within the educational systems, because we know that these are kind of critical points when children, I don't know the Canadian system, but in Austria, for instance, when they move from kindergarten to school and then from primary school basically to higher level schools, these are critical time points where we think a lot about kind of academic achievements and things like literacy and mathematical abilities, but social well-being and connectedness in different social environments, the family, peer relations, and teacher-learner relations are things that we should think much more about, and where I think this sort of research, hyperscanning research, can really give us some new insights. Mental health is another aspect. It's not my focus of research, but I like this quote by Leo Schilbach that psychiatric disorders should be seen as disorders of social interaction, where I think we can do a lot for patients and also improve relationships between clinicians and patients. And then finally, digitization is another kind of big, big change or big challenge or task that we're faced with. I think this research can help improve and facilitate human-machine interaction. We can kind of learn from human interaction how human-machine interactions can work. But at the same time, this is something that I pointed out with some colleagues in a recent perspective paper. We should always strive for a balance between, on the one hand, human need for sociality, and on the other hand, system transparency, because otherwise we end up with machines that are superhuman-like, but underneath it all, not really human-like or don't have human or animate qualities. So, and this is something that children in particular are very susceptible to. So they are very kind of susceptible to things like social appearance, faces, eyes, social responsivity, and then develop strong empathy, for instance, for machines that might not be warranted always, and might even conform like show increased social conformity to machines that we might not want. And then, of course, yeah, remote virtual human interactions, something that, as we experience now, is also something that could probably benefit from a better understanding of the mechanisms of how people communicate, and could perhaps, yeah, be improved in this regard. And I think this is it. Yeah, I'm thankful to my wonderful team at the University of Vienna. Again, please check out our homepage and Twitter if you're interested. Thankful to our funders, and also thank you for your attention.
Thank you for this wonderful talk, and it was super clear, very interesting. So we'll take the questions here in the room, but also for people at home. So please raise your hand if you have a question for people at home. So we can start. Yes, there's a question.
Yeah, I don't know if I'm like well heard from here.
Can you hear
Yeah, I think so.
Okay. So I was wondering kind of, because we know in the history of psychiatric disorder, we have like a long story of kind of putting the burden of the disorder on the mother. And I was wondering, in this new modern like era of research, how do we make sure that we don't go back to that of putting the fault on the mother or the caregiver?
Yeah, that's a very important point. Especially, I think, in autism research, right? There was this kind of cold mom idea that the mom is unresponsive or not affectionate enough. And actually, the very first video that I showed you of the still face paradigm that Adronic developed, the idea was kind of to simulate a postpartal mom with postpartal depression, who sometimes show kind of flat affect, but of course this kind of total still face is not something that a caregiver would naturally produce, even with postpartum psychopathology. Yeah, we now understand much better that there's lots of variability also in how postpartum depression actually manifests in the interaction with the infants. A lot of these moms don't show any kind of aberrant interaction patterns with their infants. Yeah, so this is one, I think, important insight. And then we kind of see a subset that does show kind of flattened affect, flattened expression, but others are actually super engaged and almost, again, intrusive. So I think looking at interactional patterns, understanding interactional dynamics is super important. But as I also try to always convey, it's important to understand that it's not a one-way street in either way. Yeah. So I think what becomes very evident from this interactional research is that the kind of onus of engaging in the interaction or keeping it going is never, even in the very young infants that we are observing, is never only on the caregiver or only on the infant. Yeah. Instead, what we see is these patterns of mutual engagement that seem to be crucial. And yeah, I think taking this interactional stance should actually help show that, or make an argument for, or illuminate the dynamics at play here. That it's that yeah, it takes two to tango, so to say. Yeah. So what we're seeing here is that the infant, as much as the caregiver, is actually engaged in keeping these interactions going. And perhaps something that I didn't mention in the talk, but what we're also trying to do is involve more fathers also, because it's also something that's important, that it's quite even though it's sometimes called the traditional family, where it's basically the two parents and the child, and the mom is taking care of the child all the time, we know, of course, this is not traditional at all. It's very unusual in human history, where usually, alloparenting takes place, where lots of different people take care of children. So what we're trying to do is also understand better the interactional patterns that fathers, for instance, show with babies, where we also see neural synchrony, and we also see it especially in face-to-face interactions, so not so much different from the moms, but we see different behavioral correlates, which I think is also super interesting. And I'm hoping we will be able to look at grandparents as well, interacting with infants, but yeah, so much that we should do to understand actual social experiences that infants have much better. But I think this research will also show that, it's not all up to the mom. Yeah, on the contrary, infants are engaged in social interactions and social relations with different people and at different levels.
I have a small, two quick questions. Thank you so much for your presentation. Can you hear me?
Yes.
Okay, good. One for the spatial more, so is it the same regions that you see for the mom and the infant that are coherent, or you just did across the brain? So this one. And second is more about theory of mind, because you showed a temporal parietal junction in one of the slides. So I was wondering if you did like a follow-up, like 3-4 years old, when there is like already a theory of mind, and if you're planning to do that, if it's something you'll be interested in.
Yes, thank you. So in the FNIRS hyperscanning studies that I showed you, we always looked at homologue regions. Yeah, so basically we looked at regions of interest. I think, it was temporal TPJ, basically left and right, and then frontal regions left and right, and we only looked at homologous regions, showing this coherence. Two limitations with that. So one, that of course with the FNIRS system, at least with the system that we have a limited number of channels, so we don't cover the whole cortex. Of course, with FNIRS, we don't cover deeper structures at all, so another limitation. And then only looking at homologue regions, I think, it's sort of straightforward, but it also might, we might miss a lot of information, because I think a lot of the time, especially when we're thinking about communicative exchanges, it intuitively makes sense that when I'm assuming the role of the listener, it's more like auditory or perhaps theory of mind regions that are engaged, and more motor regions, speech motor regions in the speaker, for instance. Yeah, so I think it makes a lot of sense to also look at kind of heterologous regions, but we haven't done that here. And then in terms of perspective taking or theory of mind abilities, we haven't tested that directly in our studies so far. I think it makes sense as soon as we're kind of moving in this territory of more higher level conceptual alignment, which we haven't done so far. So I don't think this is something that's needed for kind of the lower level alignment in terms of affect and these communicative rhythms that might be at play in proto-conversations even. There's infants won't at 4 to 6 months, when we showed these proto-conversation effects, as even their receptive vocabulary is so limited, they won't understand a lot of what's going on in the conversation on the parents' part, right? So I don't think this is necessary for these kind of lower levels of alignment. But once we are moving into the direction of, higher level conceptual alignment, like the storybook reading task this is a project that I'm planning to do there, I think this will definitely play a role.
Hi, can you hear me?
Yeah.
Yeah, okay, thank you. Thank you so much for the talk. It was really interesting. My project is actually on Fnir's hyperscanning in mother-children diet as well. And I have a bit of a like specific question.
Okay.
We're wondering like what periods, like what epochs to select when conducting
Oh
our
God.
yeah.
Oh God. What is your task?
So we've got a free play, like 10-minute kind of interaction, and we also have
Right.
snack time where they freely talk and play. Sorry,
Okay.
eat, but there's a lot of artifacts, so we don't really have like a straight like 60-second that's clean. So I was wondering in your
Mm.
study, in the conversation studies, like do you select long periods, short periods, like random periods, or select them? Yeah.
Not completely random, no. So yeah, we don't have a task structure. It makes it difficult, especially in the free conversation, free play. So in the puzzle task, actually, well, we looked at roughly how long it took them to solve one puzzle. So that would kind of structure the task a little bit, but there was lots of variation. So not really. And then in the free conversation, yeah, there was no task structure at all. So we basically looked at a very broad band of frequencies. I would have to look into the paper to tell you exactly. But we didn't restrict it much to say, well, it's more conservative if we take a rather broad frequency band. But it's a recurring issue or question. I don't think anybody has found a kind of gold standard method to, or, you know, if you look at the literature, I think there's also across papers some variation in the frequencies that were selected. So yeah, actually, I mean, if you're interested in getting a little bit into an exchange about the topic, a postdoc of mine, Carolina Pletti, is looking at that right now in a data set that she collected. So perhaps that's useful just to get in touch and have a little bit of conversation about that issue. Because, yeah, it's...
Thank you.
It's great. Make collaboration like this. Okay, so do we have another question? No, maybe I have one. Do you compare modalities and like neural synchrony? Is there a modality where you can find like better neural synchrony? I'm thinking about like comparing hearing like language and like sign language. If we compare it, is there a difference or?
Mm-hmm. Okay. I'm thinking I'm not aware of the study that has directly compared different modes or channel of communication, to be honest. I would have to think about it. What I can say so far is that we see these entrainment effects on the one hand and the synchronization in different modalities. Yeah. So and it's kind of the ability of the brain to detect regularities and to pick up on rhythms and to kind of show these resonant responses. This is something that we see in different modalities. Yeah, we see it visual and auditory and tactile. And one thing I can say that we know quite well also from adult research is once the information is multimodal, it's easier to pick up. So this is kind of one suggestion or one idea why it's so helpful to actually see mouth movements when you're listening to someone speak, because it's just another modality that's redundant. Yeah? And we also see that it's possible to entrain brain rhythms in one modality, and this enhances the processing of stimuli that are then presented in phase in another modality. So this is the only thing that I can immediately think of where we see an advantage, yeah, if it's multimodal. But per se, I mean, if it's just speech, just auditory, or just gesture, yeah, sign language would be super interesting to study. I'm not aware of a study looking at that, but I think it's also rhythmic. I would be very much surprised if kind of rhythm didn't also play a role there, kind of in, but I don't know, maybe you have experts on sign language.
No, but I
there
was
that,
super interested because
yeah.
like studying deaf individuals and like before I post the cochlear implants. So that would be super interesting to see.
Super interesting, yeah.
Okay, well, thank you so much. Do we have another last question maybe in the room or maybe at home? No? Okay. Oh, we have one last.
So we're very interested in the FNIRS hyperscanning results that you were showing, and I was wondering whether they're consistent with the EEG hyperscanning on the same task. So in other words, when you see increases or decreases of synchrony, do you see something consistent, or would you expect to see something consistent with EEG?
Yeah.
How do we move forward if different modalities give
yeah.
How do we interpret that?
Yeah, that's also an excellent question. So yeah, I think I mentioned it only briefly. So as soon as I switched to the hyperscanning, I also switched to FNIRS in my talks in the studies that I presented. Just because we've had some trouble with EEG hyperscanning, especially with the infant, and it starts with the movement artifacts, especially when we're performing tasks that require vocalizations. They are very hard to get rid of in the EEG. So these naturalistic tasks where we have lots of back and forth and behavior that we're actually interested in, so we don't want to get rid of it, that gets very, very tricky. So basically the only EEG hyperscanning study, and then of course we're looking at different rhythms, right? So we're looking at these very slow oscillations with the FNIRS linked to the bold response. And then we're looking at EEG rhythms at a much higher frequency, which would be super interesting in principle to understand better during social interactions. But then we have, again, the problem with the artifacts. And then we have the problem, the conceptual or methodological problem that these rhythms actually differ between the infant brain and the adult brain, because infant brains are wired differently. They have slower oscillations that we think have the same functionality as the adult rhythms. So lots of trouble actually trying to do EEG hyperscanning with infants or children and adults at the same time. The one study we did was actually also with a frequency tagging paradigm, because I thought, well, let's just give them a rhythm artificially, basically, but we didn't find synchronization there. Perhaps again we were too far off from a natural interaction then, or we didn't look at the data the right way yet. And then now we have one study where we collected data during a musical interaction, EEG data from mothers and infants. So I'm curious to see what we will find there. But yeah, to come to your kind of more conceptual question, would I expect to find the same conditions to induce synchrony or the same behavioral correlates? Per se, yes. On the other hand, I think we also need to much better understand neural oscillations. This is a part of our research that I haven't talked about today at all. We're doing really basic research into the development of neural oscillations in infants. And so in FNIRS, when you're looking at these slower oscillations, and then, yeah, practical difficulties when actually trying to co-register. So we ran one study actually with adults where we looked at FNIRS and EEG at the same time. So far, we only managed to really analyze the FNIRS data in a way that I'm confident in the EEG data was too noisy. So we've so far only published the FNIRS data from the task. It was a kind of simple rock paper scissor task. We do have EEG data from that task, but haven't managed to look at it yet. So once we've managed that, I can tell you whether we see the same patterns. I'm not aware of a study, actually published study, co-registering EEG and FNIRS in a hyperscanning paradigm and publishing those. If you're aware, I would be very interested, but I don't think anybody's managed. Very tricky.
Can I ask you a quick follow-up question to that?
Yeah, sure.
Synchronization, well, drop in synchrony that you showed during, I think these were the complex tasks such as tickling, the longer stimulation with the mother and infant. There was a drop in synchrony. In the other conditions, though, there was an increase in synchrony, if I remember correctly. And I'm wondering whether the drop in synchrony is also partly explained by the tickling, the stimulation that is, there's more movements, it's more complex, maybe the ability, even maybe the signals are not as clean. Could it be explained by logical issues? That's my question.
That's a good question too. So it actually, the data were collected in the same condition. It was a free play interaction. We just saw a kind of a linear relation between the duration of the affectional touch. So that was positively related to the neurosynchrony. And we saw a kind of a negative relation of the duration of tickling and the synchrony. So I wouldn't even call it a drop because in that study, and that's again, we're running into the limitations of FNIRS actually, because what would be interesting would, of course, to look more closely at the unfolding dynamics during the interaction, to see whether in response to the touch, do we see an increase, and in response to the tickling, do we see a drop? Yeah, let me put it this way. In the kind of feasibility study that we ran with to see whether we could do neurofeedback, we realized that we would need to measure about 50 seconds to get kind of a good estimate of the level of synchrony if we're using wavelet transform coherence, which I think makes sense for other reasons, to kind of give a useful feedback to the diet. Yeah. So to have a better temporal resolution with FNIRS and with wavelet transform coherence is again something that's difficult. But if we're accepting that this is just something that we kind of see on the group or level of the level of the whole duration of the interaction, yeah, it might actually play a role. So if I'm just imagining a situation where there's lots of tickling, lots of kind of intrusion, lots of motion, that might affect the signal quality in the way that it turns synchrony down. Yeah, I mean, of course, in the FNIRS, again, we're less susceptible to movement artifacts and we're monitoring signal quality and we're cleaning the data. So I don't think that would fully drive the effect, but I cannot rule it out completely that it had an effect on that negative correlation. It's a good point.
Thank you so much. It was amazing. And we wish you were here with us to continue the conversation on a glass of wine. But
yeah, that would be nice.
Maybe next time. And it was really a pleasure. Thank you so much.
Thank you so much. Thank you also for the nice discussion. And yeah, have a nice evening. Bye-bye.